This is a production of Cornell University.
Thank you for coming today. I'm really excited to talk to you about what we've been working on up in Geneva with Willow. And if you get a second, please check out our website. We've now coined the term Willowpedia. And if you have any further questions after the talk about Willow, you can find out some more information on the website. The Energy and Independent Security Act of 2007 basically stated that we want to try to reach 36 billion gallons of renewable fuels by 2022. Currently, if you look over here, currently our status of cellulosic biofuels is pretty minimal compared to this graph and compared to other conventional biofuels. And those consist of ethanol from corn and sugarcane. And those are expected to not increase that much within the next 10 years and actually will eventually plateau off. Whereas cellulosic biofuels are going to increase, and they're going to increase significantly. And we need to start looking at other crops other than corn and other than sugarcane in order to reach the goal of 36 billion gallons by 2022. So back in 2005, the USDA and the DOE conducted a study, and they called it the billion-ton study. And the point of this study was to determine if the United States had enough resources, biomass resources, in order to replace 30% of petroleum usage by 2030. And that was back in 2005. And they actually found out that we needed to produce 1 billion tons, dry tons of biomass yearly in order to replace that 30%. Back in the spring, they just put out an update to the billion-ton study, and this figure was taken from that. And basically showing that by 2022, we should be able to produce over that billion-ton mark. And if you notice, this orange is energy crops. And along with an increase in pretty much all of these resources, forest land resources, logging residues, forest product residues, along with the current agricultural crops that we are using, we're going to have to increase energy crops in order to reach these goals, and actually to even reach our billion-ton limit. Okay, so what are energy crops? They're dedicated energy crops. They're not crops that are going to be used for food. So corn is an example. It's used for food, and we also use it to make ethanol. These would be strictly used for bioenergy. Some of these are Miscanthus giganteus and perennial grasses, prairie grasses, such as switchgrass in this picture. Others are canary grass, big blue stem. There's a bunch of different types of prairie grasses that could be that have the potential to be good energy crops. What we're looking for is something that can produce high amounts of biomass in a short period of time, and these can do it. In addition to the grasses, we also have woody bioenergy crops, and included in this are hybrid poplar and also shrub willow. This is a picture of Fred right there, Larry's new grad student. This is image was just taken about a month ago, and at a trial up in Geneva, and that's about 3 years growth right there. Okay, so we have all these different types of feedstocks, and the problem is that multiple universities from all around the country are working on different feedstocks, studying them for yield and other pest and disease resistance and other specific traits. And so the Sun Grant Initiative was set up by Congress to basically work together to create a national network where we can combine all of this research that's been going on all these different crops and all these different trials. And they divided the United States into the 5 different regions, and basically came up with the Regional Biomass Feedstock Partnership. And they set up centers at each of those regions. And for the Northeast, Cornell is that center. Okay? And most of their funding is coming from the Department of Transportation, the USDA, and the Department of Energy. In addition to this national network, there are also the bioenergy research centers that have been set up by the Department of Energy. And those are located in California, up in the Great Lakes, and down in Tennessee. So essentially this figure is showing you that the United States is working on developing renewable energy, and it is forming some form of a cohesive national connection. And so this is a rather complex image, but what this image is showing you are the field trials for all of the feedstock crops throughout the United States. And this isn't all of them, this is just a representation of a decent amount of them. The blue stars represent poplar, and you can see that they're scattered pretty much around the United States, the upper Midwest, the north, and in the south as well. If you notice, the black stars are willow, and they are mostly within the Northeast. So the idea is that the feedstocks would be regional, because the ideal growing conditions for a particular crop are not going to be all over the United States. They're going to be in regions of the United States. And for willow, that would be the Northeast. Okay, so a little background on shrub willow. You can find, they are members of the genus Salix in the Salicaceae family. And you can find native and naturalized species throughout the region, along riparian and wetland habitats, along the edges of rivers and streams, and in any clearings, edges of railroads and roadsides. They are a pioneer species. They are dioecious, which has, so they're obligate outcrossing, and they've maintained high levels of genetic diversity as a result of that. Many species can hybridize, and they have a short generation time, and they propagate clonally, which is rather advantageous from a breeding perspective. In addition, I do want to talk about a little bit about the cultivation, the history of the cultivation of shrub willow in the United States. Actually started out for basket making, and this particular species, Salix purpurea, was brought over from Europe for the purpose of making baskets. And probably in the early 1900s, the basket making industry with willow kind of declined, and so a lot of these fields of willow, Salix purpurea, were abandoned. And now Salix purpurea has naturalized throughout the region, and you can find it in wild habitats, similar to this picture right here. So for our purposes, we want to cultivate willow, but we want to cultivate it for a bioenergy crop. And so we have to go to an unnatural setting, to a field setting. And in order to do this, we have to prepare sites and basically kill any weeds with herbicides such as Roundup, and then also apply pre-emergence before we actually plant. And this image here is an image of our zone tillage, we first set this up this year. We're trying zone tillage and comparing it against conventional tillage. Conventional tillage is where you till the whole field, whereas in this case, you're only tilling the rows where the willow will get planted. And this is a great image to see that we plant in double rows. Okay. So we plant with unrooted cuttings. You take willow whips, and they are fed into a cutter head. They're cut to about 30 centimeters, pushed into the ground, and then these packer wheels pack the dirt back around that willow cutting. And there's an example of some of the cuttings that we would plant. Gives you an idea of how the size of the cutting. And there it is in the ground. And then by the end of the summer, so say we plant in May or June, by the end of the summer they'll look like that. And that's not very big. They dedicate most of their energy to root production in the first year, so they don't grow very well the first year. So that's why weed control is extremely important. These guys can easily get overgrown by weeds if the weed management isn't taken care of that first year. That winter, the plants are cut back to the ground, and then in the spring they'll resprout, and that's the regrowth in the spring. After a year growth, we're here, and then 3 years after coppice, we will then cut the willow. Okay, we will essentially harvest the willow, and this cycle can continue for about 7 years, roughly. And you can harvest on about a 3 or 4 year rotation. Some people can even do a 2-year rotation. So it just depends how often you want to harvest. And the 7-year harvest cycle, this limitation is not mainly due to growth of the willow. The willow will continue to grow vigorously, but the stools get too large and it's harder to get equipment through. So we have to actually, it's time to pull out the stools and replant. Okay, so for harvesters, we actually have quite a few different harvesters that you can work with, depending upon your needs and practices. There's these guys on either side here, which are rather large. They're heavy, but they're great because they can cut those double rows at the same time. And but they do use up quite a bit of energy, and they are very heavy. So for larger field trials, they'd be great. But for smaller field trials, it might be rather inefficient. So we've have this. These are modified forage harvesters, and this one can do 1 row at a time. It's a little bit smaller. It can be attached to the back of a tractor, and it's easier to get through for smaller trials, and it'd probably be a little bit more energy efficient. And then we also have the option of a bio baler, so you can actually bale the willow. And also you can harvest the whole stems. So if you wanted to harvest for planting stock, since we plant those willow whips, you don't want to actually chip the biomass. You want to keep those willow as whole stems. So those are some options for harvesting. So what do we do after we harvest the biomass and we have a bunch of wood chips essentially? There's multiple options for biomass conversion. There's basically 2 main types of conversion. You've got thermochemical and you have biochemical. Thermochemical conversion includes basic combustion, which is just burning the biomass, or co-firing it with coal is another option, and that'll help reduce the emissions for the coal plants. You can densify the willow into pellets and briquettes, and then burn via combustion. And then you can also undergo further processing through gasification or pyrolysis. This will produce syngas, biochar. And then you can go through biochemical conversion, which is essentially accessing the polysaccharide component of the biomass, converting it into monosaccharides, into sugars, and then fermenting those into ethanol or butanol. And you can also use this to make other bioproducts. For us, there's multiple phenotypic traits that we are interested in looking at. First and foremost is yield. We want to produce a big producing crop. That's our product. The biomass is our product, so we want a lot of it, and we want to be able to do it cheaply. And one issue that we do have are some pests and diseases. Insect damage and fungal pathogens can have a significant impact on yield, so we need to make sure that we are, as we're breeding for high-yielding willow clones, that we are also maintaining some level of pest resistance and disease tolerance. This is an image of willow rust that can have a significant impact on yield. And in addition to that, we are also looking at woody biomass traits. Density and composition in particular. And this is going to really help evaluate the quality of the biomass and also allow us to identify what genotypes can be marketed for the different types of conversion processes, the thermochemical or the biochemical. So again, the main goal is high yield, and that's mainly been captured through hybrid vigor. And this diagram just shows you a nice, gives you a nice idea of the species that we've worked with and the diversity that we've maintained within the breeding program. And also the hybrids that do work and the ones that actually don't cross very well. The dotted lines are basically no seeds are being produced through those. We do work with about 20 to 30 different species within the breeding program, so it is a larger array of species than a normal agricultural crop that might have 2 or 3 species. Okay, so we're going to talk about biomass composition today because that's the main focus of my talk, and woody biomass traits. And just a quick review that biomass is essentially the reflection of the cell wall chemistry, and the cell wall in a woody stem mainly consists of 3 main polymers: cellulose, hemicelluloses such as glucuronoxylane, and lignin. This image here is actually of a primary cell wall. So you do have pectin in there as well, along with the cellulose microfibrils. And there's no lignin in this cell wall because it hasn't penetrated in, and the pectin hasn't been dissolved and the lignin hasn't taken over. But it still gives you a pretty good idea on how complex the cell wall is. And the cell wall's designed for structural integrity. So, and barriers to pathogens for the plant. So it's actually not easily broken down. It's very recalcitrant, and to access those polysaccharides is not an easy process. And especially for the biochemical conversion side of things, we need to understand a little bit more how to break down the cell wall. And so there's been a lot of work being done on that. But my focus is more on composition. Can we impact, by changing the composition, can we actually impact conversion and improve the conversion process, or improve the pre-treatment process and access those polysaccharides better? But first, we actually have to be able to characterize the biomass in a rather efficient way. The wood composition, traditionally, wood composition is characterized by old wet chemical methods. These are based off of the TAPI methods, technical applications of the pulp and paper industry, and also the newer methods by NREL, standard biomass analytical procedures. And these require harsh chemicals. You're using a lot of equipment. You are boiling toluene, boiling ethanol, boiling sulfuric acid, basically to pull out the polysaccharides, separate out the lignin, and be able to actually quantify those components within the wood. And it's rather time consuming. And so the newer processes that we're looking at are instrumental methods, actually developing chemometric models to determine composition. Although we do need the traditional wet chemistry analysis in order to calibrate any of the newer models that we are trying to develop. I'm going to talk a little bit about near IR and TGA. This image over here is an IR spectrophotometer, which I will actually be starting to test some samples on within the near future. I have done some work on developing near IR models. And then I've also done some work with this instrument over here, which is called a thermogravimetric analyzer. The advantage of the spectroscopic techniques, the IR and the near IR, is that this is a non-destructive method. Essentially, you just grind down the biomass and you can analyze it with total reflectance, and then you're not losing any of your biomass. Thermogravimetric analyzer is actually burning the sample. So you do end up losing it in the process. And it does take a little bit longer than the IR and NIR models. This is an image of a thermogram of a sample that was put through the TGA. And what I want to point out here are the two curves. You have a black curve, which is essentially weight loss with respect to temperature, and then you have the derivative of that curve, which is the blue line. The thermogravimetric analyzer works it's pretty straightforward. It actually just burns the sample as it weighs it. Now, this classically has been used to resolve polymeric mixtures. As long as each individual polymer within the mixture has a different decompositional temperature. As long as they combust at different temperatures, you can resolve them. And essentially, wood is a mixture of polymers, so that's how this works. You start of this dotted line here, right here, represents water loss from the sample. And then as you increase in temperature, you will start to combust hemicellulose. And then as you continue, you'll start to combust cellulose. And cellulose, the crystalline cellulose peak is right at 300 degrees. So crystalline cellulose burns exactly at 300 degrees Celsius. And then following that, you will lose a little bit more cellulose and you'll start getting into lignin combustion. Now this is done in complete air. If we were doing this in a deoxygenated environment, that would be pyrolysis, and you wouldn't get complete combustion of these three components, and you'd end up with char at the end. Okay. The other advantage of doing the thermogravimetric analysis is that there's been a lot of work of using this to study the kinetics of wood and looking at thermochemical conversion processes and understanding a little bit more about the thermal decomposition of biomass. So I have developed three models, one for cellulose, hemicellulose, and lignin, based off of using wet chemical data and correlating that to the results from the TGA. So the x-axis is the true value, percent cellulose determined by wet chemistry, and the y-axis is the predicted value, the percent cellulose determined by the TGA. And ideally, you want a one-to-one curve on this. So you really want y equals x. So you really would like to see an r squared of 1, but that's not going to ever be the case. And so these were the best models that I could come up with. For cellulose, an r squared of 0.74. For hemicellulose, an r squared of 0.86. And for lignin, an r squared of 0.76. So essentially, this is a pretty good model, and we can use it to phenotype a large number of samples. In addition to the TGA, I want to talk a little bit about the near-IR. This is a spectra of 3 Willow samples, 3 different Willow samples. You can tell that it's a rather complex spectra. You're not going to be able to observe specific peaks. You're actually observing the overtones of absorbances of bonds, not the actual absorbance of the bonds like you would in infrared. The advantage of NIR over infrared is that you don't have to deal with too much water absorption. You do over on this end. There's a little bit of water absorption coming in over here, but it's not as much of a problem as doing IR spectroscopy. So this really requires a very good chemometric model to resolve these spectra and actually identify what changes in the spectra are correlating to changes in composition. So I started working on this a couple years ago, and the issue is that the NIR needs a larger calibration set than the TGA does. So a lot more samples that have been characterized. So it does take a little bit more time. This graph over here is predicted versus true. So predicted is on the x-axis. True is on the x-axis. Predicted is on the y-axis. And this one is cellulose. The green represents bark samples. So just the bark has been stripped off of the stem. The orange is the straight biomass. So that's the whole stem, wood and bark. And then the blue is just the wood. So the bark was removed and it's just the wood. And these purple samples, which are only in the lignin model, or magenta, I'm not sure what you want to call that, are samples from a trial in Saskatchewan, and they have lower lignin content than any of the biomass samples from trials around here. So I threw them into the model to try to make the model a little bit better. So the models so far can distinguish these different tissue types, which is great, but they're having a harder time distinguishing genotypes from each other. So we need to work on these models better. We need to make them better, and the only way to do that is to increase the calibration set, increase the amount of samples, the known samples that you're throwing into the data set, and that's the only way that we're going to improve the model. But in the meantime, we're doing pretty good. We've got R squareds above 90%, so that's a pretty good number, but we need to be able to resolve this better. So for now, I'm still using the TGA to phenotype Willow samples, and that's it. I'm not using the NIR yet. So I put up this figure. I wanted to show you some of the things that we've been doing utilizing the TGA. I have been analyzing pretty much all of the samples that are coming out of, all the individuals that are coming out of our trials. But in this particular trial, the same individuals were planted at 2 contrasting sites. The idea was to start to begin to look at some G/E interactions. How does the environment impact composition? And this top figure is cellulose. And cellulose, based off of the stats, was significantly different by site. And the interaction between clone and site was not significant. Okay, so basically what that is saying is that at this site, Belleville, the lighter green, you will have higher cellulose content than at the other site, at the Tully site. Hemicellulose did not change that much between the 2 sites, and lignin was clone or variety and site specific. So the only 2 varieties that had significantly higher lignin at the Tully location are these two right here, and they are both Essabiminalis by Essabiana hybrids. So there still needs to be some work to begin to identify what is the actual environmental component that's impacting these. We have done soil analysis, but we haven't done enough work to actually figure out what's actually regulating these traits and how carbon allocation into the cell wall is changing based on those environmental conditions. We've also looked at ash content. So the great thing about the TGA is that after everything burns, whatever's left over is ash. So I don't have to do another step to determine ash content. So ash can be an issue because from a consumer perspective, people who have wood stoves or pellet stoves, they don't want to be cleaning out as much ash. So lower ash is going to be better for the market. Also, for the gasification, ash can cause clinkering, which can actually damage the equipment. So ideally, you want low ash, and you want it to be maintained over different environmental conditions. Depending upon where your field trials are, you want to have low ash. And you can clearly see that certain varieties have significantly higher ash at the Belleville site versus the Tully site, whereas others are maintaining low ash at both sites, like these 2 S. purpurea clones. So, in addition, I've also looked at wood density, and wood density was also significantly different between the 2 sites. It was higher at Tully compared to Belleville, and there's been some research on looking at wood density and growth rates. And there's a theory out there that higher wood density is associated with a lower growth rate. And we did find higher yield of these varieties at the Belleville site, lower yield at the Tully site. So that would make a little sense. However, there's no correlation between yield and density, and there's no correlation with yield and any of the biomass traits. Yield was only correlated with pest and insect damage. So there needs to be, again, more research, which leads me to here. We've also been doing some sugar release. So again, we thought that by altering lignin content, lowering lignin content, increasing the polysaccharide component of the biomass, that you could increase the sugar that you produced, for the sugar released for fermentation. So this is an image of sugar release from untreated biomass. So it hasn't gone through that pre-treatment process of breaking apart the cell wall. It's just basic ground up biomass, and we threw it into an enzyme mix to break down the sugars, break down the polysaccharides into sugars to see what we could get. And with these varieties, we are getting a rather wide range in sugar release. However, none of this correlated to biomass composition. So my next slide. So there are some correlations. The first one, the most important one I would say, is cellulose versus lignin. So there is some collinearity between biomass components, because as you increase the content of one, you're going to lower the content of the other, and vice versa. So it's nice to see that we can find this trend of this negative relationship between cellulose and lignin. However, this can be site dependent. So certain trials, you can't find this trend. You can't find this correlation, but other trials you can. So we still don't know what's controlling what. There still needs to be some more work. We've also found that sugar yield is correlated to density. It actually has a positive relationship with density. And there's a question of whether there's a relationship between cellulose and density. I've seen it in some trials, but I haven't seen it in others. And as you increase that crystalline cellulose inside the cell wall, that's going to impact density. So there's a question of whether or not a higher cellulose content is correlated with density, but we're not seeing that here. So, and we're not seeing a relationship between cellulose and sugar yield. So it basically, what this basically is saying, that we have a lot more that we need to learn about the biomass and how it's actually relating to sugar release, how the environment is impacting composition, and that maybe reducing lignin content, increasing the polysaccharide content, does not have an effect on sugar yield. So really, we need to start understanding the genetic component of the side of things. And we've done all these phenotypic measurements, and now it's time to start understanding the genotype. And to start, I worked on this family, S. sacchinensis by S. Vienna, the progeny of this family. So this was a cross between two parents, and we produced progeny. And that progeny we used in a study for real-time PCR. And I collected two tissue types, the shoot apex tissue and the young stem tissue, with the idea that I would be capturing primary cell wall development as well as secondary cell wall development. And I wanted to look at particular genes associated with cell wall development. This is the phenylpropanoid metabolism pathway, which leads to the production of the monolignals, the subunits for producing lignin, p-coumaryl alcohol, coniferyl alcohol, and synapyl alcohol. I looked at about 3 different genes involved in this pathway. In addition to, I'm not going to get too far into this because it's a little bit too complicated. In addition to the lignin, I was also interested in looking at the polysaccharides that are being produced in the cell wall. And in particular, cellulose synthases, UDP glucose dehydrogenase, and the multitude of glycosyl transferases that are involved in the production of the hemicellulose glucuronoxylan and also pectin. So there's hundreds of genes that are involved with the production of polysaccharides within the cell wall, and I can't go through them all. So I'm going to focus in on 2. UDP glucose dehydrogenase and cellulose synthase 4. This top figure is gene expression within the apex tissue from the 2 parent clones, SX61 and SX64, and there's 6 progeny. So the dark green and the light green are the parents, and then the in-between green are the progeny. So the top is the apex. The next figure is the stem, the lower part of the stem that I looked at. And then this last figure on the bottom is the relationship, the gene expression between the 2 tissue types. And with both of these genes, you can see that the apex has lower gene expression compared to the stem. And so basically implying that these are probably involved with secondary cell wall metabolism. So these are correlations of the gene expression of those 2 genes with percent polysaccharide content in the biomass, in mature biomass, and percent lignin content as well. And if you notice, UDP glucose dehydrogenase gene expression has a negative relationship with polysaccharide content and has a positive relationship with lignin. Whereas with cellulose, as expected, you've got a positive relationship with cellulose content and negative with lignin. So this is a little interesting question here. And there's been a little bit of work done on this by Deborah Samick at the USDA and University of Minnesota, a collaborative effort between the 2 of them to actually lower lignin content in alfalfa. And in order to do that, they lowered the gene expression they actually downregulated UDP glucose dehydrogenase. And what ended up happening was they saw the exact opposite. So basically saying that as you pull UDP glucose dehydrogenase away from cellulose, what you end up doing is increasing lignin, not lowering it. So it's kind of counterintuitive. Does that make sense? Okay, so analyzing this stuff one gene at a time can be rather difficult. So we need to start looking at this on a bigger level, on the genome level, on the transcriptome level. And even though we've started to identify candidate structural genes, we need to do a little bit more. And so that's where it comes into the sequencing of the genome and also looking at transcriptome data, and eventually leading into whole genome selection. And we started to sequence the genome. We've been working with Salix purpurea with the clone ID 94006. And we do have some preliminary data. We've done some 454, and we've done some Illumina. And we are waiting on a high-seq run. We still haven't gotten the results from that one. So our assembly so far is it's okay. It could be better. And then we also have 2 back libraries that we've created. A random shear library from Lucigen, and then a traditional restriction enzyme library from Coogee, Clemson University Genomic Institute. And we've done, with our initial assembly of Willow, we've done some comparisons to the P. trichocarpa genome. And they do have a similar size genome. And we do see a 65% of the P. trichocarpa genome aligned to that draft assembly. And the sequence identity between the coding and the non-coding regions was about 88%. So that's pretty good. In addition to that, we've done some transcriptome sequencing. We've looked at, we have 8 different tissue libraries. We collected shoot tips, RNA from shoot tips, RNA from leaves that were pooled from 3 different collection times. We did a midday collection, we did a nighttime collection, and we also collected drought stress leaves. And then we've also looked at 2 different sections of the stem. We looked at the node and the internode. We have collected roots, and we've also looked at the female catkin, because 94006 is a female. So we've looked at the transcripts in that as well. And we've also done some work with those 2 parents that were used in the real-time assays, that cross that I did. The S. saccharinensis by S. myobianus. So those 2 parents, we've also done some work with the stem and the shoot tip. These two are both tetraploid. So it'd be interesting to see the transcripts that come out of that. So our RNA-seq assembly, here's the results for the S. purpurea sequence assembly for the transcripts. If you can see, our average read length was about 394 base pairs. And based off of that, 80.5% of the poplar transcripts have at least half of their length aligned to the Willow transcripts. And the sequence identity between those two coding regions, between Salix purpurea and P. trichocarpa, is 92%. And remember, the genomic was 88%. In addition, at JCVI, they've been kind enough to create a G-browse for us. So we can now start looking at genes within Willow and essentially using poplar as a reference genome and doing a de novo transcript assembly. And the nice thing is that here's a contig, a Willow contig. Some of these contigs are actually spanning full genes, which is fantastic. And so we can not only start characterizing gene families, but we can also start looking at some expression profiles. And here is the cellulose synthase gene family from the Salix purpurea. And I compared gene expression in the shoot tip and the stem internode. And I boxed these genes that are boxed are those involved in secondary cell wall metabolism. And as you can see, their gene expression is higher in the stem compared to the shoot tip. So essentially, we've done a ton of phenotypic measurements, and we're improving our methods for phenotyping a lot of these traits. But we need to do more. And basically, what it comes down to is that we need to characterize the phenotype and the genotype better. And we'll be able to do that through using mapping populations and also doing some GBS, which we've just started to do. Fred's been working on that. And we'll be able to start doing some SNP analysis and eventually leading to whole genome selection. So we can actually start in breeding for particular woody biomass traits, which is eventually where we want to go. And my acknowledgements. Again, I want to thank Larry Smart for being my mentor for the past 6 years and providing me with financial assistance. Tim Volk and Larry Abrahamson at SUNY ESF, who are involved with the developing and getting the Willow breeding program going, well, the Willow program. Art Stepanovich at ESF, along with Larry Walker and Stefan Korgi down at the BRL, have been instrumental in the Willow, determining the composition for Willow biomass and getting those models going. And then all of the people involved with the Willow genomics at the Craig Ventnor Institute and at JGI. And then Martin Goffernay for the microscopy work. He's helped me with a lot of that. And then all my funding over the years. My current funding is coming from the Sun Grant. And that's it. Questions? Yep.
It looked like you had some interesting differences in ash content, and I can understand that you're kind of focused on the cellulosic fermentation part, but the ash might be interesting if in the future things go toward incineration or combustion.
Right.
So have you looked more at other components of the ash, for example, chlorine or sulfur,
No.
potassium, that kind of thing?
Yeah, and I've gone back in the literature to actually try to find people who've done work on actually characterizing ash in different biomass feedstocks, and that's been rather hard to find. So it should be done, yeah, because different components will impact things differently. So we do need to look at that more. And bark will have more of an impact too. more bark is going to have more ash. So we do know that. Yep.
Would you consider possible transgenic approaches to improve wood quality, or is that too difficult because of as a naturalized species and vigorous?
No, actually, there is no genetic transformation method for willow.
Got it.
We cannot do it yet. There's been a lot of work dedicated to that. Getting it into tissue culture can be difficult, but you can do it. That's possible, but you can't regenerate. So they've had a hard time on that end. You can actually, I mean, you can do the transformation on a callus, I guess, but you can't get it to actually any point. But there's still some work being done on that. So maybe someday. You think that since they're so easy to root, that you could get it to regenerate that easily, but no. I mean, people have tried different hormone concentrations, different changing hormone ratios, and they just can't get it to work. Oh dear, there was 3 right there. You can start.
How many years can you get the highest biomass?
Say that again.
How many years can you get the highest biomass?
How many years?
Yeah. I say the picture you when the third years the leaf will get very tall.
Yeah, so that's probably do you want the height? Yeah. So Larry, what's the height? Fred?
Measures.
Yeah.
Third year, third, fourth year, 20 to 25
20 to
meters.
25 meters? Feet.
Sorry.
Feet, yeah. Wait a minute. No. Yeah, 20 to 25 feet. So that's their height. And then as far as the biomass, we can get above 10 to 14 dry tons per acre? Per hectare, sorry, per hectare.
Then you harvest. And the third year you harvest the leaf.
Say that again?
Yeah, I mean when at the third year when you plant at the third.
So if you harvest after the 3rd year, are you getting that kind of yield?
Oh, yeah, no, we're getting more yield. So the next cycle of harvest, you'll get more. So the first year, you get that 10 to 14. But the second cycle, you're going to get, 20% increase in biomass production. And I don't think we know beyond that. I don't think we've gone past the second rotation.
Hey, Michelle,
Yeah,
we have a question up in Geneva.
okay.
You mentioned that you have both diploid and tetraploid, and what are the advantages of one or the other in biomass production?
Well, we've actually, most of the varieties that we've patented for yield are triploids. Does that answer your question?
No, you give me some other interesting information. You were sequencing a diploid, that makes sense because it's easier.
Yes.
And you told us about tetraploid selection that you had, but now you added more into the knowing about what you released with triploid. So
Yeah, so that initial family that I did the real-time work on, we didn't know we're tetraploid until afterwards. So we're interested in looking at that, but from a genomic standpoint, I think we really want to stick with the diploids.
Michelle, I've got a question.
Yeah.
Most of your talk is
Uh-oh.
What are you looking for in terms of actual physical size of pelletable material? Is there a restriction on size that you're looking for, a diameter of stem, or does it matter?
I don't think it really matters. The larger the stem, you're going to have a harder time harvesting. But I mean, the harvesters can handle, 3 diameter stems, no problem at the base. But, we did have this question of whether or not reducing the bark to wood ratio by increasing stem diameter, would that reduce lignin content or would that reduce ash content? We haven't done the work to figure that out yet. But as far as stem diameter goes, we have varieties that have very small diameter stems, but lots of them. And then we also have varieties that have just a few stems that are very large and thick. So, and some of those have been patented as well. So they're still producing high yield, regardless of stem diameter.
Thanks.
Yep.
Of all the varieties that are being tested throughout the nation through this fund grant, which ones have the easiest conversion of the polysaccharides extraction?
You mean the different crops?
Yeah.
Yeah. The grasses have a little bit different composition than a dicot hardwood would, but basically in general, all lignocellulosic biomass is going to be rather difficult. But I don't really know if there's been differences in recalcitrance between grasses versus the woody biomass.
So there hasn't been like a specific species that's been pulled out to say this is going to be good for creating a biofuel, this is going to be good for creating a pellet, something like that's been.
Not really, no.
Yeah.
I think in general, we're talking about just all lignocellulosic biomass, just what species or what types it will produce high yield. Not so much, more work has been on, okay, what kinds of microbes can we get to break down the stuff better, not in terms of the biomass itself.
This has been a production of Cornell University, on the web at cornell.edu.